Casey is a professor of biochemistry and molecular genetics at the University of Colorado School of Medicine. His research lab develops deep learning methods that integrate distinct large-scale data sets. And he will talk about opportunities and challenges for AI in biology and medicine. Casey, over to you.
Thank you. Yeah, so I'm excited to have some time briefly this morning to talk about what I see as kind of challenges and opportunities in these fields. I would just say I'm actually going to start with the challenges because I think the opportunities are really incredible. I mean, if we look at what happens in research and we look at what happens in medicine, there's a lot of written text, there's a lot of images, and there's a lot of cases where we need to synthesize data to learn something more. So I think everywhere you look, there are opportunities. The challenges, though, are that there's often unique features of these data that if we don't take them into account, we can end up really shooting ourselves in the foot. And so, yeah, I'll start with some challenges. So first, I really like this article from Siddhartha Mukherjee. If you haven't read it, I'd highly recommend reading it. It's about sort of it contrasts artificial intelligence and physicians. There's a quote in it from Jeff Hinton, who I'm sure that you've probably all heard now that, they should stop training radiologists right now, right? The idea is there's this fundamental advance in imaging informatics and sort of deep learning where, we're not going to need humans to look at this, at these types of images anymore. Every week, I sort of pulled things from a week back in 2019, and like everywhere you look, there were papers suggesting that artificial intelligence is basically going to replace human doctors. So maybe this is the end, right? The presentation can stop right now. We're all done. And I would say, unfortunately, that's not quite the case. There's a lot of complexities of these data where if you don't take them into account, you will build systems that appear to work well by the traditional computational metrics that we usually use, but that will lead us astray if we continue to use them in the long term. This is definitely a favorite of people on the internet, pointing out the challenge of figuring out the difference between blueberry muffins and chihuahuas. In this case, this can be fun, but there's a lot of cases in medicine where we'd be a little bit disturbed if we were mistaking two entities that were quite different. I think this Adversarial Examples that comes paper from Ian Goodfellow, where there's now a whole bunch of other articles around this that really push on the same thing. So the way I perceive this, let me just walk you through this, is I perceive this to be a panda with about 60%, and the neural network that was trained here perceives this to be a panda as about 60% confidence. We're going to add 7 1,000ths. So a very, very, very small fraction of this, seemingly random dot pattern, which, a neural network classifies with low confidence as a nematode. And what we get back is going to be something that looks drastically different, right? We took something like this, we added a very small amount of something else. It's going to look drastically different. Well, so to me, this looks about the same, right? But to the neural network that is trained here, this comes back as a gibbon with 99% confidence. And, I think this points to a hazard that, for targeted perturbations, these methods can be pretty brittle. And that hints at a challenge if we're going to use this in medicine, for instance. We really don't want the difference in the decision threshold to be this small. These adversarial patches have gone even further. So this is a toaster sticker. I know many people have probably seen this, where you can sort of place the sticker on a table and dramatically change a classifier's output. I think these types of challenges are going to be really important. Why does this actually matter for medicine, right? Why should we care, right? Those are just esoteric academic examples that are fun to look at, but maybe not that important. So let's go back to this article. This is one of the ones I sort of briefly mentioned at the beginning, a new algorithm that spots pneumonia better than a radiologist. So what we're going to do is we're going to look at a radiograph and we're going to try to see if there's sort of evidence that the individual who's had the X-ray taken might have pneumonia. So this is the image, and this is the map that's sort of asking, okay, why is this having a high probability of pneumonia? Now you note here the probability of pneumonia is only 5% according to the classifier, but that's in the top 95th, that's above the 95th percentile of sort of probability for pneumonia predictions. And you might say, okay, so positive scores here are suggesting pneumonia, negative scores are sort of saying, probably not pneumonia. If you're looking at this, you're going to be concerned about regions of density in the lungs. So you're going to be looking for sort of white patches somewhere in here. You see some of them here. You look at the scores from the neural network, those are actually negative. If you look down here at the bottom of the image where there shouldn't be anything relevant, you get these high positive scores. So whatever's happening here, it's actually, the classification is not coming from the part of the image we care about. You see the same thing here. This is an image where you'd probably be concerned in this area, but the classifier actually isn't terribly concerned in this area, right? It's concerned up here where the word portable comes from, as well as kind of other parts of the image that should be irrelevant. Okay, so what's actually happening here? Why is the system not working in the way that we think, even though from a methodological evaluation, we'd say it's working well? Well, this is a quote. I was actually reading a comment online from someone who was writing about sort of dealing with p-values, so attempts to estimate probability in high throughput data. And I was reading through the comments section of the internet, which is always a dangerous place to live. But I got to this comment. It was, on big data, data collection biases are always larger than statistical uncertainty. And I think this is like, if you are working in biology and medicine, or if you are considering working in biology and medicine, this is fundamentally a limitation. The reason those radiographs are coming back classified as pneumonia is that there are hints outside the important part of the image that suggest potential pneumonia. So if you imagine you had many different X-ray machines, some were in regions of the hospital where pneumonia was frequently seen. Others are in places where you'd almost never diagnose pneumonia. A method can learn characteristic patterns of the machine itself to identify features of that machine that are associated with like where it's positioned in the hospital. And that's not what we want these methods to learn, right? If we're going to build the next radiologist, a radiologist needs to make predictions not based on where someone was seen, but actually what's in the image. And so, for those of you who are thinking of getting into biology and medicine using these methods, or for those of you who are already in biology and medicine, I think fundamentally this is the key point. You have to think about the data. You have to think about the data collection biases. And then there's another hazard that I also worry about. So I think this is one obstacle. I think this is the biggest obstacle. But I also think this is an obstacle as we start to apply these in biology and medicine. It's really fundamental to the way that we talk about these methods. We like to say, oh, this is retrieved from Wikipedia. So this is like in image recognition. The raw input could be a matrix of pixels. The first layer learns sort of one set of features. The next layer learns a more complex set of features. And the next layer learns a more complex set of features. This is really the way we would like to think about things. It makes intuitive sense. It's perfectly explainable. The challenge is these methods really don't work like this. They sort of vaguely work a little bit like this in the first and second layers. But as you get to these high dimensional layers, they're really sort of learning very complex mixtures of features. It's not really a clean separation. There's a couple papers from Chris Olah that, again, if you're thinking about working in this field, his papers on Distill, on sort of looking at what neural networks are actually learning, are ones that you really need to think about. So, I think these are the two biggest obstacles: trying to romanticize what these methods do around sort of imagining we know how they work, and also sort of having data collection biases that are fundamental. If you can tackle both of these problems, I think you're in a pretty good place for being able to use these methods to make a big positive difference. So what should we do? So, I think fundamentally, if you're working in biology and medicine, domain expertise is going to matter a lot more now than it used to. I think the quote from Jeff Hinton at the beginning in that article, that we should stop training radiologists, is fundamentally at variance to what I've seen in the field. I think the people who work in the field are going to have an idea of what types of data collection biases exist. And by engaging with these experts, you're going to build products that actually help. If you sort of try to end run around or replace these people, I think the challenge that you're going to have is you're going to end up learning bias after bias after bias in your data. And you're going to, I think, struggle to build something that makes meaningful predictions that you couldn't just get from sort of metadata alone, if it happened to be collected. And so I'd encourage you to really deeply engage with domain experts. And then I just wanted to, I know these talks are very, very short. So, I just wanted to sort of introduce you to one way that we're thinking about how to use these methods. So, this was a project that we did with some folks at Penn, where I used to have a lab. So my lab was at Penn, and we worked with rheumatology folks. And just to give you an idea of how extensive I think collaboration needs to be, this project was a 2-year project where we really regularly sat down with the folks in rheumatology, so people who study autoimmune disease. And we said, what are the problems that you're dealing with? And how can we use technology to help? And one of the challenges that they were dealing with was they were analyzing data from rare disease patients. And what we had is basically a genome-wide snapshot from rare disease patients. And we said, what we can do here is we can try to learn from people who don't have a rare disease. We can learn what biological patterns are, and then we can apply those patterns to help distinguish features of the disease that might point to potential treatments. And really, and then they would say, well, actually, what you really need to deal with is you have to think about 2 different types of things. You have to think about sort of pathways, so how genes are expressed together, and also cell types, because autoimmune diseases involve many different cell types. And so we really had to think about how to come up with the solution to this problem. And it was a long dialogue. I just want to emphasize, like, working to advance in practice in this area is going to be a long dialogue with domain experts. And so the postdoc who led this ended up developing a method where we started with some large-scale data. We learned patterns from that, and then we could take those patterns and apply those to the rare disease setting and try to identify differences associated with the disease. And just to take you through the type of thing that we ended up seeing, and if you want to get more details on this method, it's available online. We ended up they had 3 different data sets. We took these patterns and we said, okay, which patterns distinguish disease severity in airway epithelia, in renal glomeruli, and in blood cells? So the idea was, can we find some pattern that's different in individuals who have the most severe form of this disease? I know this isn't a biology audience, so I'm not going to talk you through all the reasons that I thought the M0 macrophage signal was a really interesting finding. But essentially what we found was this cell type that is elevated in the most severe form of the disease more than it is in people who have a less severe form of the disease, and even in those people is elevated above what you see in individuals who don't have the disease. And it's systemic. So everywhere we looked, we found this. And what this did is it gave the folks a new angle to say, hey, is there a way we can modulate M0 macrophages? If this is a systemic disease and this is a real systemic difference, maybe we can find a way to tamp down the activity of this cell type and start to ameliorate some of the symptoms of this rare disease. And so, the goal of this is not really to give you a deep dive into the biology of autoimmunity, but it's really to say, look, if you have these types of dialogues as you're doing these projects, I think these are the opportunities. It's to sit down with domain experts and bring a technology to the table and really work with them to advance both our understanding of disease and methodologies. I'll say this is just the top part of our set of GitHub notebooks associated with this project. I just wanted to use this slide to emphasize, this is a long process, like multi-year to, I think, make a meaningful difference. Because the first things you always find are biases in the data. For those of you who really if you were really excited to get a deep technical overview, I think the duration of this talk didn't allow it, but I'll just say Dr. Jacqueline Taroni was the lead author of this work. The GitHub that I showed a screenshot of is also available. This bit.ly link will also take manuscript is published if you want more details. Whew. That was a fast 13 minutes.
Yes, it was really fast. Yeah. Thank you for your presentation, Casey. And it was so fast that we don't even have questions yet. Hopefully they will arrive soon. But then I'm of course not a doctor. I understand that this is a very important topic, and what you do is very important for our health and the development of medicine. But in the middle of this pandemic, can I ask you a little bit about that? So, how you see what have you seen would contribute to the COVID fight? Probably not your lab, and sorry if it's not relevant for you, but I think it's interesting for our audience.
Yeah, so I think there are certainly elements of where artificial intelligence can be really important. And areas that come to mind for me at the moment, kind of a salient point, is thinking about what do variants in the genome do? And that means both on the human side, are there variants that sort of predispose people to a more severe form of the disease? And on the SARS-CoV-2 side, are there variants that either increase severity, increase transmissibility, or evade current vaccine strategies? I think what we would all love to see is really rapid AI-based predictions around, that could tell us what the answers are going to be there. I do definitely worry that the same things that I talked about with the radiologists will be a hazard here. Biases in the data are likely to dominate the signal. However, I think the ways we can get around that are sort of approaching any of these problems or challenges with just a little bit of sort of being just a little bit humble. As kind of computer scientists, I feel like sometimes we have a chance to walk into a field and feel like a bull in a china shop, right? Like I don't know if that's a common phrase. We sort of jump into the field and sort of say, oh, this is easy. Here's how we solve the problem. And then we break everything. And I think the key is when you have, if for folks working on this, when we have variant effect predictions where you say, okay, I think this is going to make this biochemical change, being willing to be a little bit introspective in our models and say, okay, what's making me make that prediction? Is it possible that this is driven by a bias in the data, or does this appear to be real biological signal? I think if we sort of take that step back and do that analysis before we sort of, run out and say everyone should do this, I think we'll be in a better place. I worry about cases like, certain small molecule drugs that people were very excited about early in the COVID pandemic as potential treatments. I mean, we were all, I think, very hopeful that some of those would work out, especially if it was a widely available small molecule that was inexpensive. But, I think often we took our preliminary finding, what we saw was people taking their preliminary findings and just like trumpeting them immediately and sort of not going through that rigorous process of trying to understand if something worked before sort of selling it as a treatment. And I think there's a lot of lessons we can take from that. I think as artificial intelligence practitioners, if we don't do that sort of introspective step first, I think we'll reduce our credibility. And ultimately, it means that artificial intelligence will have less of an impact than it could otherwise. And I will say there are certainly some nice examples of people using artificial intelligence approaches to try to model the pathogenicity of variants. So how dangerous individual variants are in the SARS-CoV-2 genome. And I'm actually pretty encouraged by those, particularly kind of the ones that are focused on, the potential, how potentially infectious the virus will be. So yeah, I guess I would say, I think there is a role, but we just have to be careful. I think that's the fundamental thing I want to emphasize.
Yeah, careful with the virus and careful with how we approach it with AI. Yeah.
Yes.
Thank you. It was very interesting. Yes. And in the meantime, we got questions, but before we look at them, I forgot again to mention that there is a poll function just next to the chat. So it is to the audience, of course. So please rate the talk and give your feedback. And now let's have a look at the questions. So Balint asks, what do you think? When will the greater audience benefit from these kind of AI-based studies? The most interesting ones.
Yeah, so I think for those who are thinking about sort of putting this into clinical practice, I guess what I would say is, I think the first step is going to be thinking about how to augment existing practitioners, how to augment radiologists, how to augment pathologists. I think we're not terribly far from that. So it would shock me if within a decade, this wasn't kind of a routine thing that was being used. I do have some concerns about rolling that out. I think we have to make sure that the way that we're rolling that out doesn't reduce the attention that sort of radiologists and pathologists are putting into into the work that they put into each image that they examine. But I think it's going to, the transformation I think is going to start within the decade, but I hope it's an augmentation and not a replacement.
Yeah, and that helps, of course, it's spreading as well. Another question from Gergely. What methods do you propose to avoid the issues in the pneumonia example, looking at the interrelevant parts of the images?
Yeah, so I think one way that we can sort of really more reliably assess our methods is to include highly relevant baselines, like including a baseline that's just saying, if I just know the scanner, how well do I do? And I think using methods that sort of control for what we expect to be irrelevant features, could be helpful. So this is just me thinking off the cuff here, right? So take it with a grain of salt. But like I could imagine, maybe you build machine learning models of actually the scanners themselves. So you build something that can sort of take an image and put it onto sort of make it look like it was generated by a different scanner. And maybe you sort of do data augmentation where you sort of take images from one scanner and you sort of move them to others. I think there's going to be like clever strategies to control for these types of biases that we're going to need. I would say, I think we've put a lot of effort into thinking about sort of what our neural network architecture should be. And I think if we put a little bit more effort into thinking about sort of how we can control for data biases, I think we'll be more successful faster. I don't know if that's a hard and fast answer, but that's where my mind goes.
Yeah, thank you. Another question from Christoph. What do you think about black box model issues in this field?
I could answer this for half an hour. I won't, just to save the time. Yeah, if I was really confident in the data collection, and I was really confident in the model, and I was pretty sure that it was making predictions off of, features that we really think should be driving signal, and not off of sort of these biases in the data, I probably wouldn't care so much if it was black box. I guess it's a question of sort of, are we, is the goal to be an engineer and to have something that works, and maybe it doesn't matter why it works. And I would love to be in that space. Unfortunately, at the moment, just given what I've seen in the literature in this field and the products that I, have come out and sort of looking at how they've generalized or not generalized to practice, I guess I would say at this point, I think we still need to try to interpret the box. We still need to try to understand what's going on in there, simply because we need to make sure we're avoiding to the maximum extent we can these types of sort of data issues. But I would love to get to the point where I don't think it necessarily matters, but in practice, I think it actually does.
Yeah, thank you. Another question from Marcel. Could you tell us a use case where reinforcement learning can be used in medicine? If there is, how do you simulate such?
Yeah, reinforcement learning is really an interesting, it has a lot of potential promise, but it is hard to figure out how to use it in medicine. I think some sort of reinforcement approaches, where you could imagine having many examples and then only revealing certain examples, but the challenge of reinforcement learning is you kind of have to test your environment, right? It's like somehow you have to do something and then test the results from your environment. And in medicine, you could imagine some significant risks. And so, this is something I struggle with. I mean, I think it's clearly a method that sort of is remarkable in its potential power. But it's very hard to think about how to ethically use it in the specific setting of medicine. So I guess I don't have any good answers, but what is on your mind is very much on my mind as well.
Yeah. So it brings up other questions.
Yeah, it delves deeply into ethics, I think.
And questions from Gerald. Are doctors or other medical practitioners open-minded to using AI tools to assist their everyday job? And another one, don't you fear that radiologists will come and destroy medical AI?
Um, I think, there's no one group of answers with physicians. Just like there's probably no one group of answers with AI practitioners. I guess I would say, I think that certainly some people are very open-minded. I think to the extent of maybe giving us too much credit for what we've been able to accomplish thus far. So I think there's definitely some folks who are open-minded. I really think if we want to make a positive difference, we have to engage with practitioners. So in radiology, we have to engage with radiologists. If I look at sort of what happened in the sort of initial self-driving car space with companies like Uber, making a mistake that is very costly to someone's life can effectively shut down an entire program. And I think when we lack that sort of introspection and like ability to engage with experts, we're much more likely to make mistakes that are very costly and that also lead to the type of blowback that really can stop entire programs. And so I guess I would say, I think the path is going to involve sort of thinking about who the stakeholders in this process are, engaging with those stakeholders, think about how the process should be governed, who should have a say, and always keeping in mind that the end goal is to advance patient care. And so I don't think I'm terribly concerned about radiologists destroying medical AI. I think there's a lot of excitement around, having people be able to sort of maybe use AI to just make what they're doing better, right? To do it more quickly, do it more efficiently, and do it more accurately. But to do that, people have to understand what these methods can and can't do, and they have to be implemented in a way that increases physician engagement instead of decreases it.
So it's about communication, cooperation, and then everyone win-wins, especially the patients.
Yes. Yeah. I mean, if this can work, right? I mean, it's the path to, more effective healthcare. It's the path to less expensive healthcare. I mean, there are lots of wins, but if we do it wrong, it's the path to either less effective healthcare or, the sort of AI being regulated out of the healthcare space. So I think, yeah, it's very important to get the governance right.